and mute. Good evening. Dr. Sheela Turbek is a postdoctoral researcher in Dr. Kristen Ruegg's lab at Colorado State University. Her doctoral research at the University of Colorado Boulder focused on the role of mate choice and migratory behavior in generating and maintaining avian diversity. Lately, she's interested in using genomic tools to advance the conservation of threatened avian species. Dr. Turbek, we invite you to begin your presentation, and if my introduction missed anything, feel free to fill in the blanks. Take it away, Dr. Sheela Turbek.
All right, great. Thank you, John, so much for that introduction, and thank you all for being here today. It was really fun to hear about some of your recent sightings around Fort Collins. So as John mentioned, my name is Sheela Turbek, and I'm currently based at CSU, where I'm working with Kristen Ruegg. And today I'm really excited to share with you how our research group at CSU is using genetic tools to advance migratory bird conservation. And this is a collaborative effort with multiple universities, nonprofits, and government agencies that we refer to as the Bird Genoscape Project. So throughout the course of this talk, I'll delve a little bit into a few key objectives of the Bird Genoscape Project. First, I'll discuss how we use genetic information found in a single tail feather to map migratory pathways at the population level and why this matters for migratory bird conservation. Second, I'll talk about how we can also use these genetic data to investigate the potential for migratory birds to adapt to climate change. And then finally, I'll briefly touch on some of the education and outreach initiatives that we carry out to connect people with migratory bird conservation. Okay, so I'll start off by talking about how we map population level migratory pathways. And this work broadly revolves around figuring out where migratory birds that breed in northern latitudes spend the non-breeding season. And one of the reasons that this question is so important and urgent is because of recent widespread declines in native North American birds. So these figures were taken from a paper that was published in 2019, which used avian survey data to show that the number of birds in North America has dwindled really precipitously over the past 50 years, with grassland breeding birds experiencing the largest decline. And this staggering loss corresponds to about 3 billion birds that have disappeared since the 1970s. While the study found that some avian groups are being affected more than others, migratory birds as a whole are experiencing some of the biggest population declines. And so the Bird Genoscape Project focuses specifically on neotropical migrants, or birds that breed in the US and Canada and then migrate south of the US border to overwinter in Mexico and Central and South America. While we often think of these species in terms of their behavior on the breeding grounds, in reality, they spend over half of the year south of the US border. So because these birds are traveling between breeding and wintering sites on an annual basis, it can be difficult to actually pinpoint why these population declines are occurring. Migratory birds are subject to numerous stressors that occur across the annual cycle in disparate geographic areas of the world. So these stressors include habitat destruction, predation by house cats, diseases like avian influenza, which has been in the news a lot lately. And then on top of all this, they have to cope with the impact of both current and future global climate change. And so it's unclear where these birds are actually most threatened. They could be declining due to events that are occurring on the breeding grounds, at migratory stopover sites, or on their wintering grounds, which are often located thousands of miles away from where those birds are actually breeding. And so in order to determine how these regions are linked to one another and develop effective conservation strategies, it's really essential that we identify migratory connections between breeding and wintering sites. For example, if habitat destruction is occurring on the wintering grounds of this yellow warbler, knowledge of migratory connections between breeding and wintering populations will allow us to link this habitat destruction back to population trajectories on the breeding grounds in the US and Canada, and determine which breeding populations are likely to be affected. In the past, these migratory connections were largely identified through bird banding efforts. And these efforts began in North America in the 1920s with the formation of the North American Bird Banding Program, which was designed to track and monitor migratory birds. And while this program has been successful at identifying the migratory pathways of particular avian species, it really has its limitations in small-bodied songbirds that are in at least the ones that our group works on. So in fact, overall recapture rates for songbirds away from the site where they were actually banded are only about 1 in every 10,000 banded individual. And this corresponds to a recapture rate of about 0.%. So with that kind of recapture rate, you can imagine how long it would take to accumulate enough resightings to reconstruct population-specific migratory routes, particularly in declining species of conservation concern. More recently, a number of new exciting technologies have been developed that provide information about various levels of avian migration. So for example, Birdcast is a really cool collaborative project between the National Audubon Society, the Cornell Lab of Ornithology, and a bunch of other organizations that uses radar data to provide real-time predictions of bird migrations. If you check out their live bird migration maps, you can actually watch the real-time movements of migratory birds as they travel in mass across North America, which is pretty incredible. You should definitely check this out if you haven't already. And this kind of tracking data provides insights at the level of all birds, because radar doesn't allow you to distinguish particular individuals or populations from one another. There have also been incredible developments recently in tracking technology that provide insights at the level of the individual. So for example, the miniaturization of direct animal tracking tags like geolocators and GPS devices now allows us to gain information about individual variation in the timing of migration and migratory routes. And while these technologies have their advantages for answering particular questions about avian migration, the Bird Genoscape Project specifically aims to fill this knowledge gap of how to track migratory birds at the population level, which is something that remains challenging to do using some of the previous tracking methods that I mentioned. And to convince you that tracking migratory birds at the population level is actually important, I'm going to use the example of the willow flycatcher, this rather unassuming and drab insect-eating songbird that breeds throughout the continental United States and a little bit up into Canada. The willow flycatcher has been separated into a number of different subspecies across its breeding range. And prior to our work on this species, very little was known about population or subspecies specific migratory patterns. However, these subspecies have been declining at different rates. So in particular, the southwestern willow flycatcher, which ranges from Southern California east to New Mexico, has plummeted since the mid-20th century, likely due to a number of different factors, including habitat loss, water diversions, and urbanization. This subspecies was listed as federally endangered in 1995. So while information on the migratory behavior of all of these 4 subspecies is important, pinpointing the migratory pathway and wintering location of that southwestern subspecies in particular would allow us to better understand the stressors that might be causing its population decline and develop more effective management strategies to help protect it. And in order to do this, we need to generate a population-specific map of migratory flyways for each of these 4 subspecies. So our approach for creating this map uses high-resolution genetic tags that are developed from DNA contained in the tissue at the base of tail feathers. This DNA allows us to identify a unique genetic code that corresponds to each breeding population, which we can then use to trace the breeding origin of individuals that are captured anywhere along their migratory journey. This approach has a number of different advantages. So first, it provides information on population-specific movement patterns, and it can be applied to pretty much any species of bird without the need to recapture individuals, which, as I mentioned, is a limitation of many other tracking methods. In addition, the feathers that we need for these analyses can be collected with minimal impact. Dropping feathers and molting is a normal aspect of avian biology. So this is a much less invasive strategy of tracking migratory individuals than fitting birds with animal-borne tracking devices that they have to carry all the way down to their wintering grounds and back. This approach can also be used to ID individuals that are subject to collisions with things like wind turbines and cell phone towers, poaching, disease outbreaks, and even mass mortality events, and determine which breeding populations those individuals belong to. So we're actually currently working on acquiring samples of the Wilson's warbler that dropped dead in a massive die-off event that occurred in New Mexico a couple of years ago, in order to determine the breeding origin of those individuals and why that die-off event may have occurred. And then finally, this project has the potential to connect biologists and citizen scientists from all across the Western Hemisphere on a really massive scale, since we rely on collaborations with a wide variety of organizations and banding stations in order to acquire the number of feathers that we need to reconstruct population-level migratory routes. Okay, so using our example of the willow flycatcher, I'll now walk you through the process of going from the DNA and feathers to actually generating a map of population-specific migratory patterns. And in order to do this, we leverage some new sequencing technologies to scan the genome for particular genetic variants. So in the past, and specifically for this species, the willow flycatcher, we used what's called a reduced representation sequencing strategy, which sequences only a fraction of the genome or set of genetic material from an individual at high coverage. And that resulted in about tens to hundreds of thousands of genetic variants that we would detect. More recently, we've switched over to low coverage whole genome sequencing, which allows us to look at the entire genome or the entire set of genetic material from each individual and results in millions of genetic variants. From these millions of variants, we then identify unique genetic tags that are representative of different breeding populations. And then we use these informative genetic regions to generate a map of genetic variation across geographic space. And this is actually what we refer to as the genoscape. So here's what this final map or genoscape looks like for the willow flycatcher. And in this particular case, we generated this map using 502 individuals that were sampled from 52 different locations across the breeding range. And in order to create this beautiful map, we basically run a statistical program that analyzes differences in the distribution of genetic variants among populations, and then identifies the most likely number of discrete genetic clusters or breeding groups. So here, the black dots that you see across the US show the breeding locations where we sampled individuals of the willow flycatcher. And then in this plot at the top of the map, I'm showing the results of that statistical program, where each vertical bar in that plot represents the genetic makeup of a single individual that was included in that analysis. So for example, all of these vertical bars that are depicted in orange correspond to individuals from the endangered southwestern subspecies. And you may notice that this program identifies 7 genetic clusters in the data, which correspond to 7 distinct breeding populations. So we have the Pacific Northwest shown in green, the interior west in purple, the east in blue, and then the Southwest, which correspond to those 4 described subspecies. And then the program also detected some additional genetic variation near the Kern River in California, Southern California, and then White Mountain in Arizona. So once we determine the number of breeding groups and we've created this nice map of genetic variation across space on the breeding grounds, we can then use the DNA from feathers to assign migrants and overwintering individuals that are captured outside of their breeding range back to these distinct breeding clusters. And this is a part of the process that requires the collaboration of a massive number of nonprofit organizations, government agencies, and academic institutions to obtain the number of samples necessary to reconstruct migratory routes. Here, for example, you can see the distribution of bird monitoring stations on the breeding grounds in blue and the winter grounds in red, many of which we've collaborated with over the years. So this effort was started by Tom Smith at UCLA and is now largely maintained by Kristen Ruegg at CSU. And it's resulted in a really impressive collection of about 250,000 feathers from approximately 100 bird species. And these feathers were collected across a 25-year time series. As you might imagine, keeping track of all of these feathers, which are sent to us from all across the Western Hemisphere, is a really massive effort. So I'd just like to recognize the work that our lab manager, Christine Rain, and then all of the undergraduate researchers at CSU do to help maintain this collection. Once we receive a feather sample, the feather sample is assigned a unique identification number. The metadata for that feather is carefully entered into a database, and then it's organized with other samples from that species, and the catalog feathers are then stored in a freezer where the DNA contained in the feathers will remain viable for an extended period of time. And then in order to actually assign the feathers from migrants and overwintering birds back to breeding populations, we follow one of two processes depending on the type of project that we're working on. So one option is to extract the DNA from the feathers and then do what is called SNP genotyping, where we basically come up with a panel of 96 informative genetic variants that we know differentiate our different breeding populations. And then we sequence each individual at those particular regions in the DNA sequence to assign individuals captured outside of the breeding range back to our different breeding groups. Some advantages of this approach are that we can screen approximately 800 feather samples in a week. So it's a pretty fast process. And it works pretty well with low quantity DNA. And that's a limitation when working with feathers that don't contain a lot of genetic material, especially for small-bodied songbirds. Another option, which we've been doing more frequently lately as sequencing costs have continued to drop, is to again extract the DNA from each feather sample and then perform low coverage whole genome sequencing, which again provides information at every position across the genome of an individual. Some advantages of this approach are that it generates much more data for subsequent analysis than the approach I just mentioned. And it also works well with low quantity DNA like that found in feathers. It can also now be comparable in price to the previous option, depending on the number of samples that we end up including. So going back to our breeding ground genoscape for the willow flycatcher, we've now sequenced an additional 363 wintering individuals and 580 migratory samples in order to connect overwintering locations to breeding populations and get the overall genoscape map. So here, the green dots that I'm showing you represent individuals that were captured outside of the breeding season that were assigned back to that Pacific Northwest breeding population. And you can see that the range of these samples goes all the way down from Western Mexico to El Salvador. And the migratory samples that were assigned to that breeding group were primarily detected along the Pacific Flyway. Next, we have the interior west population in purple, which was found to overwinter in El Salvador all the way down to Ecuador. And the individuals from that breeding group were detected along both the Pacific and the Central Flyways. The eastern population wintered from Panama to Ecuador, and these birds were detected along the Central, Mississippi, and Atlantic Flyways. And then finally, we identified that endangered southwestern willow flycatcher overwintering in specific locations in Nicaragua and Costa Rica. So knowing where this certain subspecies spends the winter then allows us to pinpoint particular wintering areas that should be prioritized for protection and carry out more targeted conservation efforts. We can also use this information that I just showed you in order to gain insight into the timing of migration in each of these populations. So in this animation that I'm about to show you, each dot is a bird or feather that was collected on a given date. So we can see how these birds are actually migrating through time. Let's see if I can start this here. Okay, so in May, you'll see that the southwestern willow flycatcher begins to arrive on its breeding grounds while our other subspecies are still migrating through. Then these birds are remaining on their breeding grounds for the duration of the summer until around September, when you see that they start to migrate again. And then you'll notice that there's this hole in the data from about October to November when samples weren't really being collected. And then in December, the interior west birds start to arrive in southern Mexico and Central America. And then finally, in mid-January, we see the appearance of that endangered southwestern subspecies on its wintering grounds in Nicaragua and Costa Rica. So just kind of another cool way to visualize the data. So the overall goal for this part of the Bird Genoscape Project is to work with NGOs and agencies to create genoscapes for as many species of as we possibly can. We're currently working on about 30 species in collaboration with our many partners, and these genoscapes are in various phases of completion. But one of our objectives is to map migratory flyways for 100 species of concern, ideally by 2025. Here's just an example of some of the genoscapes that we've already completed for a number of different species. So we have the common yellowthroat, the Wilson's warbler, and the yellow warbler on top, and then the common loon, willow flycatcher, and American kestrel on the bottom. And once we've generated maps for a variety of different bird species, we can then begin to compare these maps and search for any commonalities in terms of spatial patterns of population decline to try and get at some of the factors that could be driving the decline of North American birds more broadly. So in this first part of the talk, we were focusing on how to track migratory birds at the population level. And hopefully I've convinced you that screening feathers using high resolution genetic tags is a really good option for addressing this question. So now I'll tell you a little bit about some of the other things that we can do with the resulting information from this approach. So in the second part, I'll discuss how we can also use these data to investigate the potential for adaptation to climate change in migratory birds. If any of you had the opportunity to go to the talk that Randall Boone gave back in March, you'll already be somewhat familiar with the ways in which climate change is affecting bird populations. But a study published in 2020, which assessed the vulnerability of 600 foreign North American bird species to climate change, found that over 2/3 of North American bird species are either moderately or highly vulnerable to climate change with an increase in mean global temperature of 3 degrees Celsius. And unfortunately, that temperature increase is expected by 2100 under a business as usual trajectory. So climate change is threatening many aspects of avian biology, driving changes both in the timing of reproductive activities and seasonal migration. And in order to respond to warming temperatures, birds can either shift their geographic ranges to escape changing climatic conditions, or they can remain in place and adapt to climate change by evolving changes in their DNA sequences. So we've been using data from the Bird Genoscape Project to examine that second potential response, the ability of birds to remain in place and adapt to changing environmental conditions. And this is work that was primarily done by Rachel Bay, who's now a professor at UC Davis on the left, and Kristen Ruegg, a CSU professor and the co-director of the Bird Genoscape Project on the right. And for this section of the talk, we'll stick with our now familiar friend, the willow flycatcher. So before I joined the lab, Kristen Ruegg was interested in evaluating how populations of the willow flycatcher are being affected by climate change. And in particular, which populations will need to adapt most in order to keep pace with changing environmental conditions. So to investigate this question, she obtained environmental data from publicly available databases for each sampling location across the breeding range of the willow flycatcher. And these data included information on a number of different variables, everything from temperature and precipitation to elevation and tree cover. And so first she had to figure out which components of climate actually matter for the willow flycatcher. And to do this, she carried out a series of association analyses to determine which of these environmental variables are most strongly associated with genetic variation across the breeding range. And then she used these results to identify particular genetic variants or variable regions of the genome that were associated with those important climate variables. So just to give an example of one of these associations, this plot shows genetic variants or particular regions of the genome that varied among populations that were strongly associated with mean temperature of the warmest quarter, one of the climate variables that she analyzed. And the strength of the association is shown on the y-axis, while region in the genome is shown on the x-axis, with the different colors indicating different chromosomes. Each circle in this plot here indicates a particular genetic variant and its association with mean temperature of the warmest quarter, with anything above that dashed line indicating a significant association with that environmental variable. In addition, the red stars on the plot indicate particular genes that were located close to these strongly associated genetic variants that are known to play a role in thermotolerance in birds. So you can see that some of these genetic variants, like this one that I've circled here, are really strongly associated with mean temperature of the warmest quarter and are located near genes that are known to be involved in thermal tolerance, which suggests that these genetic variants likely play a role in climate adaptation. And if we take a closer look at this genetic variant that I've labeled with a triangle here, climate 20, you can see that there's a positive association in this case between the frequency of that genetic variant, which I'm showing on the y-axis, and mean temperature of the warmest quarter, our environmental variable on the x-axis. For example, in the desert Southwest, where this environmental variable tends to be highest, the frequency of that genetic variant is also high. Okay, so once she identified current associations between the environmental variables and genetic variation across the range of the willow flycatcher, such as that association that I just showed you, she then used future climate projections to predict these relationships into the future and assess the degree of genomic vulnerability, or the amount that populations are expected to have to adapt in order to keep pace with climate change. And when you plot out genomic vulnerability across the breeding range, this is what you get. So here, red indicates high genomic vulnerability, or areas where we see that the greatest mismatch between genes and the changing environment, while blue indicates low genomic vulnerability, or areas where populations won't have to adapt very much in order to keep pace with climate change. And the subspecies boundaries of the willow flycatcher are shown with the black lines. So when Kristen compared estimates of genomic vulnerability among these 4 subspecies of the willow flycatcher, she found that Extimus, the southwestern willow flycatcher, had higher estimates of genomic vulnerability than the other 3 subspecies. And this finding reflects both past population declines and current population abundance data in the willow flycatcher. So as you'll notice in this map on the left that shows patterns of population abundance, abundance is really low in Southern California, Arizona, and New Mexico, where the endangered southwestern willow flycatcher is found. So overall, the study indicates that the southwestern willow flycatcher will have to adapt most in order to keep pace with climate change, and is therefore at the greatest risk of climate-induced extinction. And we think that the willow flycatcher may be especially at risk due to climate change because it breeds throughout the desert Southwest, which is predicted to experience more frequent heat waves as global temperatures continue to rise. In addition, it also relies on riparian habitats along streams and waterways that has dwindled over the past century, but is thought to provide refuge from some of these extreme temperatures. So overall, Kristen's findings are really concerning and leave the future of the southwestern willow flycatcher uncertain under a scenario of future climate warming. And one of the reasons for this uncertainty is because we don't have a great understanding of the pace at which we can expect organisms to adapt to changing climate conditions. And this was a question that I was interested in addressing with willow flycatchers when I joined the Bird Genoscape Project last year. So specifically, how quickly can we expect birds to adapt to climate change? So this research project is very much still a work in progress, but in order to address this question, I'm sequencing museum specimens of the willow flycatcher that were collected in the late 1800s and early 1900s prior to the onset of significant anthropogenic change. And I'm comparing them to modern samples from across the breeding range to search for evidence of climate adaptation that's occurred over the past century. So far, we've generated whole genome sequences from 225 contemporary breeding samples from across the breeding range, shown here in black, and 17 museum specimens that were collected during the breeding season near San Diego, California from 1988 to 1903, which I'm showing in orange. And our preliminary analyses show some really interesting changes in the distribution of genetic variation among populations of the willow flycatcher from the past to the present. So I'll show you this result through a plot called a principal components analysis, or PCA, which is often used to visualize genetic differences among populations. And this analysis basically transforms a really large set of variables, like information at each genetic variant of interest in the DNA, into 2 variables, PC1 and PC2, that capture most of the variation in the data set. So just to orient you to what this plot is going to look like, in this PCA plot I'm about to present, each point represents an individual, and the closer our populations are to one another in this PC space, the more similar they are genetically. On the other hand, if our populations form separate clusters, they show genetic differences. Okay, so here's what that PCA actually looks like for our contemporary samples of the willow flycatcher using whole genome sequencing. So the points in this plot are colored by the geographic location where these birds were sampled. And you'll immediately see that this plot pulls apart three main groups. So we see the core range of the southwestern willow flycatcher circled here in red, the eastern subspecies circled in blue, and then everything else kind of clustering together. And I'd just like to point out that our contemporary San Diego samples, colored with that gold color, are clustering right about here where that arrow is. However, when we add in those museum samples from San Diego onto this plot, which are shown with the gold triangles, you can see that they form their own cluster all the way up on the top left of the plot. So this provides some intriguing evidence that a lot of genetic change has occurred in that San Diego population from the late 1800s to the present day. And in order to link the changes that have occurred in this area specifically to climate change, we're currently following a workflow very similar to the one that Kristen followed to investigate genomic vulnerability. So first, we are assessing which environmental variables are associated with genetic variation in our modern samples. Then we're identifying particular genetic variants that are strongly associated with those environmental variables. And then finally, we will compare the frequency of these genetic variants between the contemporary and historical samples to determine if they've changed over time, as we would expect in order to adapt to a warming climate. So to go back to that previous picture of DNA sequences from a number of different individuals, if we found that having a G rather than an A at this particular region in the genome was associated with higher temperatures, we would expect the frequency of individuals that have a G in the population to have increased over the last 100 years in accordance with climate warming. So in the second part of the talk, I discussed some of the ways that we're using high resolution genetic information from feathers to gain insight into the potential for climate adaptation in wild populations. For example, this information can be used to examine genomic vulnerability to climate change and assess extinction risk in the future using future climate projections. We can also look into the past by sequencing museum specimens to investigate whether organisms are already adapting to a warming climate through changes in their DNA sequences. So we've covered these first two sections, and now I'd just like to briefly mention some of the education and outreach initiatives that we run through the Bird Genoscape Project. One of the really unique strengths of this project is that it has allowed us to build partner networks throughout Latin America, since collaboration with researchers working on the wintering grounds is really essential to build the feather collections that we need to reconstruct migratory pathways. And one of the ways in which we've sought to build capacity on the ground and collaborate across international borders is by offering training workshops in genomic sequencing techniques and analyses. So this is a picture that was taken at a genomics workshop that members of the Bird Genoscape Project organized in Mexico City in 2018. And we held a similar workshop in Mérida in Mexico this past January, which unfortunately had to be held online due to the ongoing pandemic. We also plan to hold another workshop in 2024 at CSU's sister campus in Todos Santos in Mexico. Since I started my position with the Bird Genoscape Project, we've also partnered with the Environmental Learning Center at CSU to conduct local outreach with Title I schools that support economically disadvantaged students in the Poudre School District. So we've designed an educational experience specifically for middle school classes that teaches students the importance of migratory bird conservation. This program has been a ton of fun to put together. It includes interactive stations on binocular use, mist netting, and bird banding, and then a number of fun games focused on migratory behavior. We held this event with a number of school groups last fall, and we're planning to work with some additional middle school classes this year. And then finally, the Bird Genoscape Project has partnered with National Geographic and Days Edge Production to discuss the work that we're doing to advance migratory bird conservation and document how this project is connecting researchers on the breeding grounds, like Mary Whitfield, who studies the willow flycatcher in the Kern River Valley in California, with researchers on the wintering grounds like Natalie Sanchez Ulate, who's studying migratory birds in Costa Rica. Many conservation efforts in the Western Hemisphere focus on the breeding grounds of migratory birds, which are typically in the US and Canada. But in order to adequately protect these birds all the way across their annual cycle, we really need to be communicating with conservation practitioners that are working on the ground in areas where these birds are passing through and spending the winter. So facilitating these connections is a really important aspect of the Bird Genoscape Project. And if you want to learn more about this project and some of the media outreach that we've done, you can check out this beautiful video called Feathers in Flight that is available on the National Geographic YouTube channel. Okay, so hopefully I've convinced you that a single avian tail feather contains a ton of useful information that can be used to advance migratory bird conservation, from mapping population-level migratory pathways and evaluating the potential for adaptation to changing environmental conditions, to connecting researchers working on similar species in different regions of the Western Hemisphere. And that this work is important for protecting the incredible diversity of migratory birds that we enjoy observing both here in the Front Range of Colorado and throughout North America. So with that, I'd just like to introduce you to some of the wonderful members of the Bird Genoscape team that work to generate the various genoscapes that I showed you today. I really can't stress enough that this is a collaborative effort on so many levels between our research group here in Fort Collins, our affiliates at UCLA, and our collaborators in Mexico. And I have a lot of people that I'd like to thank for the work I presented on today, particularly the Ruegg Lab at CSU and the Smith Lab at UCLA, both of which work to maintain the giant feather collection that I mentioned. And then, of course, the thousands of biologists who have contributed samples that make projects on such a massive scale possible. And then last but not least, we wouldn't have been able to carry out any of this research without the support of numerous funding agencies. So particularly the National Science Foundation, the Department of Energy, the Department of Defense, and then both National Geographic and UC Mexis, which provided funding for some of our conservation or our outreach initiatives. And then finally, I'm really grateful to Fort Collins Audubon Society for giving me the opportunity to speak today. So thank you so much for your attention, and I'd be happy to take any questions.
Just a second. Great, Sheila, that was terrific. And first I want to double check. Can you hear us again on Zoom?
I can.
Okay, great.
Yeah.
Before we before I open up to questions, I'm going to take moderator's prerogative. Sheila, how much space does 250,000 feathers take up?
Yeah, that's a good question. So they're all in the freezer, and I think actually I'm not sure how much space it's taking up, but in that the National Audubon Society actually covered the work that our lab is doing in the recent magazine, and apparently it's 15 freezers or something that we're storing feathers in. It's kind of insane.
Thanks for that. So now the floor is open for questions. If you're on Zoom and you have a question, type it in the chat window. We will get to it. Here in the room, please raise your hand. And I think instead of handing around a mic, I'll just repeat your questions. I saw your hand up first, Ron, and then Joe, and then if anybody else. Go ahead, Ron, first.
Yeah, could Sheila put up back on that screen that showed the National Geographic film?
Yeah.
Yes, they can hear your question sometimes.
That was like, thank you.
Oh,
Yeah, it's about 15 minutes long, I think.
I don't think this is a question. I think Ron just wanted to see.
Just
the
wants
name,
to,
so-
yeah.
No.
Thanks.
Joe, it looked like you had a question.
Okay, how big is the bird genome versus a human genome?
Oh my gosh, yeah, it's much smaller. So birds are actually great to do genomic studies on because they have pretty small genomes. So it would cost a lot more to do something similar with humans or mammals in general.
We got a question from Zoom. Andrew, what is it?
So Liz Brewster says, what an amazing project. How many labs do the feather DNA analysis? How much longer will this project continue?
Sorry, can you repeat that?
How many labs are participating in this, and how much longer are you funded to do this work, or do you expect to do this work? Which Liz thinks is fantastic.
So as far as how long, I guess as long as we can continue to receive funding, we're all really passionate about the work that we do. And then in terms of number of labs, so our main collaborators are here at CSU and then at UCLA, but we have all sorts of people at nonprofits and government agencies that will have feathers that they want analyzed by us. And so we'll collaborate with them on various aspects of these projects. For example, if there's a die-off event and like US Fish and Wildlife collects a bunch of samples, they might ask us to try and ID those individuals to figure out which breeding populations they were from. So we collaborate with a ton of different organizations.
Any more?
Robin Welsh wonders, will any feather work, or why do you use tail feathers?
Why do you use tail feathers?
Yeah, presumably any feather would work. The tail feather is, so it would be harder to get, I think it would be harder to get DNA from contour feathers, just like small body feathers. So we take tail feathers because it's less invasive than taking something like a primary or a secondary, which is needed for flight. And so just plucking one tail feather, it doesn't get in the way of the bird's normal activities as much, and still contains the information that we need.
Exactly on that. It sounds like you partner with bird banders, and when the birds release, sometimes they do a stress mold. Can you use those feathers and then tie that bird to the band? You have, all that information. Would that be something?
Yeah, certainly. If there are feathers that they opportunistically collect, yeah, but they will definitely send them to us. And banding stations are great to work with because they have all of the associated information for the birds that they're capturing. It's a good question.
Sheila, I'm curious if your research can also contribute to work to, if it can also tell you whether birds are speciating. I don't know if that's the correct scientific term. Or in fact, we know that there's always these lumpings and splittings. I would assume this work contributes to those arguments about whether two closely related populations that are currently considered one species are actually two, or vice versa.
Yeah, so it can certainly, contribute to those discussions. And it can provide insight into any kind of like migratory divides that there are across the species range. So maybe on one side of a divide, they're migrating in a particular direction, and in the other side, they're migrating to a completely different location. So it can, give us some insight into how these birds are reproducing with one another. And that's actually kind of my background. Those are the kind of questions I studied for my PhD.
Any other questions? Yes.
Are there any of the species that you're finding that it's good news that they're not endangered?
That is a good question. I specifically was interested in working on a species where there were conservation issues at stake. But I believe there are some species. I mean, there certainly are. We are kind of trying to focus on species of concern just because that's where we want our resources going towards right now. But for example, we have a map for the yellow warbler, which is declining in some areas, but as a whole is doing fairly well, I would say.
So one that has lots of subspecies, I think,
Mhm.
or populations. Other questions? Any online, Andrew?
Well, it just came in, so I'm going to see it. Great presentation and fascinating work from Michelle. If any feather will work for your analysis, have you thought about citizen projects similar to iNaturalist or eBird? If somebody finds a feather, they can send it to the lab with location found.
Yeah, that's a good question. So we do end up storing the feathers in a freezer in order to make sure that DNA in the feather is viable. So I'm not exactly sure how long or how quickly you'd have to get the feather to us and store it in order to be able to extract meaningful genetic information. But that could be a kind of fun project if it was possible.
They can always sequence it at home, right?
Yeah, right. Hopefully one day. Sorry, I'm sitting in the dark now. I'm going to turn on my light real quick.
I've got an iPhone and an internet connection. How hard can it be? Another question for you, Sheela.
All right, Nick Komar wonders, are willow flycatchers actively differentiating or diverging genetically, or are they converging?
That is a wonderful question and something I'm interested in following up on. So we found that in the past in San Diego, the population looks very different genetically than it does now. And so I imagine that's because of some kind of hybridization or gene flow between what used to be the southwestern willow flycatcher and some of the other subspecies, but that's something we're curious about. And I don't have the answer yet.
But on the chart you showed, Sheila, wasn't that one population, the historic records were more isolated from clusters than the current records?
Yeah, exactly. So they looked more distinct genetically than they do now, which suggests that they could be interbreeding with some of the other subspecies.
Yeah. All right. Any other questions here in the room? What about online? All right. I think that does it. I do have one announcement to make, but before I get to that, let's show our appreciation to Dr. Sheela Turbek one more time for a fantastic presentation.
Thank you.